Hi everyone, I'm Dr. Kevin Hagedorn, and I'm here to talk to you about a technology which has been developed by our startup company located at Techtown, Detroit, which uses carbon surfaces to isolate RNA with single molecule purity. In 2003, it was discovered that single-stranded nucleic acids could bind carbon surfaces. It's also been known for some time that carbon is relatively inert and is used in medical implants because it doesn't biofoul. It was our idea to try and create a product which could bind single-stranded nucleic acids and isolate them from a solution with extremely high purity. With support from the National Science Foundation, we first performed simulations to determine the optimal carbon surface chemistry, and then engineered a method for adjusting the sp2-sp3 ratio and surface charge of carbon such that we could isolate RNA specifically on a carbon surface with single molecule selectivity. Shown on the left is a transmission electron microscope image of the magnetic beads that we create, a metal core coated by this carbon, and on the right is an image of our actual product. With funding from the Michigan Economic Development Corporation, we performed a study comparing our product to Fisher Mirvana and Kaigen RNeasy, two popular competing products that you may have used in your lab. What we found was that for Mirvana and Kaigen's RNeasy, there was still significant DNA contamination, which had to be cleaned up with DNase, as I'm sure you're familiar with. However, with our product, even without DNase cleanup, there was no detectable DNA contamination up to 40 PCR cycles. Furthermore, the yield was about 20% higher, and the RIN value was significantly better. The RIN value was better because DNase cleanup also reduces RNA integrity. This is a known phenomenon. If you'd like the full workup for other tissue samples and the protocol used to determine these results, contact me and we'd be happy to provide you with documentation. Another benefit is that this method and this carbon surface can be used to stabilize RNA. Shown on the left is the carbon-coated magnetic beads mixed into a solution. On the right are the beads after they bound RNA. They form these aggregates which protects the RNA from degradation even if left at room temperature. So shown on the right, we've left the RNA bound in these carbon bead aggregates for 25 days and even dehydrated the sample and rehydrated it with no loss in RNA integrity. In summary, we've developed a product which is very efficient at isolating single-stranded nucleic acids from solution. And what single-stranded means depends on your buffer conditions. So for example, with a buffer that doesn't open up the secondary structure of RNA, you'll only get short RNA. With a buffer that opens up the secondary structure, you'll get total RNA. With still further buffers that denature DNA, you can get total nucleic acids. So it's a very flexible product that can be used in multiple applications, and it will perform these isolations with basically single molecule selectivity for whatever you're looking for. This eliminates the need for DNA treatment if you're isolating total RNA. It's excellent for low sample volumes because there's no background binding, which is a problem with other magnetic bead-based products. It's compatible with Trizol, which is a popular reagent for RNA isolation, and it's cheaper than major brands with higher purity. We're located at Techtown, Detroit, which is about a mile away from the Wayne State University School of Medicine. Particularly for universities in the state of Michigan, we're more than happy to visit, demo the product, and in general help you get the most out of your samples and minimize the costs. Thank you very much for your time. Below is our contact information, and don't hesitate to reach out if you think you might be able to use our services. Thank you.